Hello students, I hope you are preparing your NEET exam with consistency and hard work. Don't waste your time, don't get confused, and don't doubt yourself. In this video, we are going to cover the revision part of the examination. These videos are enough for you to score nearly above 620 marks, and especially when it comes to biology, it is enough for you to score 360 out of 360 marks in this NEET examination. This is our third video, and in this one, we are going to study about human reproduction and reproductive health. It is a complete exam notes. Every line of this video has been selected from the NCERT, and those lines which are high yield and which are exam oriented, I have selected only those points. And also every previous year question from 1990 to 2025 has been integrated with the concept, and exactly this concept is being taught for you. High scoring video, this is the only video series you need for your examination. अगर आप री नीट एग्जाम लिख रहे हैं तो ये एक वीडियो सीरीज आपके एग्जामिनेशन प्रिपरेशन के लिए काफी हो जाएगा. कोई और नोट्स आपको ढूंढने की भी जरूरत नहीं पड़ेगी. Hopefully, let's move on. So, topic one, the male reproductive system. Okay, so in this reproductive system, I need not you explain you about reproduction and types of reproduction and how that reproduction has evolved with time and with evolution. I'm going directly to the topic which is directly in the textbook so that examination point of view revision can be done. Okay, so we are going to study both the reproductive system, that is male and a female reproductive system. Okay, the main parts when it comes to male reproductive system is testis, scrotum, testicular lobules, and seminiferous tubules. Stop the video, read this multiple times. Okay, let's see about scrotum. Okay, see, I have curated these images in order to make you understand easily. One, and also I believe that the more you see the content, the more you imagine, and the more you have image-based explanation of the same lines, it will be easier for you to understand, retain, and also recollect during the exam. So let's see scrotum. You can see the image I have curated specifically to understand why the testes are located in the scrotum part. What is the exact reason? Okay, so the body temperature is 37 degrees centigrade, whereas the scrotum temperature is 34 to 35 degrees centigrade, and it is almost 2 to 2.5 degrees centigrade lower. That lesser temperature when compared to the body core temperature is essential for the production of sperm. The production of sperm is called as spermatogenesis. अगर टेस्टिस बॉडी के अंदर होते तो बॉडी का जो कोर टेम्परेचर है वो टेम्परेचर के वजह से ये स्पर्म्स प्रोड्यूस नहीं कर पाता. इसीलिए टेस्टिस को जो है ना स्क्रोटम आउटसाइड द बॉडी इन ए हैंगिंग बैग सैक लाइक स्ट्रक्चर जो है ना यहाँ पर एनाटॉमिकली पोजीशन है. सो द रीजन इज essential for spermatogenesis is temperature. Compared to the body core temperature, the scrotal temperature is 2 to 2.5 degrees centigrade lower. That is the reason you have to understand true or false statement or assertion reason statement can be asked. Testis structure. Let's see what are the parts of a testis. You have to see the diagram. That diagram is very important. The moment you understand the parts, then you will understand the flow of semen, because the flow of semen is asked as a previous question, from which part to the which part, what is the flow pattern is seen in the during intercourse. That type of sequence questions are asked, so you have to remember the testis part here. Okay, spermaticode, you can see here, this is called as spermaticode. Okay, and that goes into dense covering, it is covered. Okay, then the outer layer is obviously the scrotum. This oval shaped structure is testes, and inside that one you can see this epididymis. Okay, so this is the oval organ, you can see the shape here, and it is 4 to 5 centimeter length. The length of the adult testis is 4 to 5 centimeter, and it is 2 to 3 centimeter width, and it has nearly 250 testicular lobules. Okay, now we are going to study the lobule organization. So in one testis there are nearly 250 testicular lobules. So what are testicular lobules? See, one testicular lobule has one to three coiled seminiferous tubules. So in one lobule there are one, two, three. Sometimes it has only one, sometimes it has two, sometimes it has three. So one, two, three coiled, it means it is convoluted, it is coiled seminiferous tubules. It is a site of sperm production. In the seminiferous tubules only the sperms are being produced. So we are studying scrotum, then we have studied testis, and in that testis there are 250 testicular lobules, and each lobules we are seeing there are one to three coiled seminiferous tubule. Each lobule has one to three coiled seminiferous tubules, and these seminiferous tubules are the site of sperm production. Direct fact-based question is been expected. So scrotum, low temperature, testis, 250 lobules, 1 to 3 seminiferous tubules in sperm production. This is high yield, very frequent previous year question has been seen. Sometimes diagram based questions are also seen, and this is an NCRT line to line points. Remaining all those introductory points or those points which are commentary in nature, which are explanatory in nature, I have removed all this one so that directly before the exam you can study this one easily. Moving on, previous year questions. Okay, let's see the question first. The testes in humans are situated outside the abdominal cavity inside a pouch called scrotum. Okay, the purpose served is obviously the sperm production. Outside the body the temperature is nearly 2.5 degrees centigrade lower. Okay, so why outside the body? Spermatogenesis requires a temperature 2 to 2.5 degrees centigrade lower than the normal body temperature, the core temperature of the body. If temperature rises, sperm production decreases or may stop completely. So answer is to maintain the scrotal temperature lower than the internal body temperature necessary for spermatogenesis. Okay, so you have to remember is no cooling means no sperm production. If there is no cooling, there is no sperm production. And what is the key point? Lower temperature in scrotum is essential for spermatogenesis. NCRT fact is direct point here. Okay, testes lie outside the abdominal cavity in the scrotum to maintain a lower temperature necessary for spermatogenesis. Temperature maintained, that difference is very important. Okay, this is examiner's favorite point also. Moving forward, cells within the seminiferous tubules. Let's see what are the cells present inside the seminiferous tubules. We have studied in our previous slide also. Each testis has nearly 250 tubules and lobules, and each lobule has 1 to 3 seminiferous tubules, and this seminiferous tubule is a site of sperm production. So this seminiferous tubules has different type of cells. Let's see, there are three types mainly. Okay, one is male germ cells, spermatogonia, and the second is Sertoli cells, also called as nurse cells, and third one is Leydig cells, that is interstitial cells. So these are the three types of cells. You can see the diagram also. Sometimes diagram based questions can be asked. So male germ cells are called as spermatogonia, and the Sertoli cells are also called as nurse cells, and other third category is Leydig cells. See, let's see the location and function. Stop the video here and see this image for 2 minutes and read the whole image for 2 minutes, and you can see directly, you can compare the type of the cell, the location and the function. Such a comparative table is easy to understand, and also this image based study will help you to retain your knowledge and also to recollect during your examination. So the location of the spermatogonia male germ cell is inside the lining of the seminiferous tubules. Function of these male germ cell spermatogonia is to undergo meiotic divisions to form sperms. So these cells undergoes the meiotic divisions to form sperms. So the function, location and type sometimes match the following questions, or multiple statement true or false type of questions could be asked. So you have to remember these things properly. Sertoli cells also inside lining, it provides the nutrition to germ cells regulated by FSH, that is produced from the pituitary gland. So it has an extra point here. Okay, Sertoli cells also called as nurse cells. They are also present inside the lining. The function of these Sertoli cells is to provide nutrition. That is why Sertoli cells are also called as nurse cells. And the production of the Sertoli cells is regulated by a hormone which is produced by the pituitary gland. So Sertoli cells production is regulated. It is maintained. यानी ज्यादा होना है कि कम होना है, या फिर स्टॉप करना है कि प्रोडक्शन कंटिन्यू करना है. That type of regulation of the Sertoli cell production is influenced by the pituitary gland, which produces a hormone called as follicle stimulating hormone. Okay, this FSH, follicle stimulating hormone, has an importance in the ovum production also, in the female reproductive system also, and also it has an importance in the sperm production also. So it has an indirect role by means it is helpful to nourish or give or provide nutrition to germ cells by Sertoli cells. That is how the FSH is going to regulate the production of Sertoli cells. You have to remember the role of Sertoli cells directly on the germ cells, function of the germ cells to produce sperms, and the role of Sertoli cells which influenced by the FSH produced by the pituitary gland. Moving on to Leydig cells, okay? The Leydig cells also called as interstitial cells. You know the word interstitial means tissue, okay? So these cells are interstitial spaces. They are present in the interstitial spaces outside the seminiferous tubules. So we have studied that location of the germ cells is inside lining, and the spermatogonia, and the location of the Sertoli cells, nurse cells, is also inside lining, whereas the location of the Leydig cell, that is interstitial cells, is the interstitial spaces outside the seminiferous tubules. The function of these Leydig cells is secrete androgens, a male hormone testosterone, under the LH stimulation. Now, comparative questions that may make you confused can be asked. Sertoli cells influenced by the FSH produced by the pituitary. Similarly, here Leydig cells influenced by the LH stimulation, that is luteinizing hormone stimulation produced by the pituitary. Okay, so the function of the Sertoli cells is to provide nutrition, and the function of the Leydig cells is to secrete androgens, testosterone, in for the uh, sperms. Okay, these androgens are produced by Leydig cells. So this table is enough. Stop the video here and observe the table and compare the points. Okay, you can see the diagram: spermatogonia, Sertoli cells in between, and sperm cells are developing here. These outside tubule cells are Leydig cells. This is the transfer section of uh, seminiferous tubules. Quick recall: spermatogonia, meiotic divisions, sperms. Sertoli cells nourish and support germ cells, FSH. Influenced by FSH, Leydig cells testosterone secretion, and it has a LH dependent because it is stimulated by the LH production by the pituitary. Key points again: spermatogenesis occurs in the seminiferous tubules. Sertoli cells are also called as nurse cells because they provide nourishment. Leydig cells are also called as interstitial cells because they are present in the interstitial spaces outside the seminiferous tubules, and FSH has a stimulating effect, regulating effect on Sertoli cells, and LH has a regulatory effect on the Leydig cells. Both FSH and LH are produced by the pituitary gland in the brain. The memory trick is FSH feeds for the factory, that is Sertoli, and LH leads the lab, that is Leydig, to make the testosterone. Okay, let's move. Male accessory ducts. So apart from the main structures of the testes, there are some ducts in the male reproductive part. That ducts are very, very important. Okay, and especially the correct order of that duct is important. You have to memorize the correct order of these ducts. In the examination, he is going to ask you direct these order of the ducts, and could be sometimes a harder question to recollect and answer, or it is easily confused. So let's see the sequence of the these male axillary ducts I have given in a diagrammatic format. Just look at their seminiferous tubules inside each lobule. Okay, so first sequence from inside to outside. First step is seminiferous tubules, then retate testis. We are going to see that what is retate testis, and then vasa efferentia, then followed by epididymis, followed by vas deferens, ejaculatory duct, urethra, and urethral meatus. Sometimes examiner is also going to confuse with some English grammatical sentences like followed by and is following. There is a difference there. Okay, order is very high yield need question. He can ask all the sequence in A, B, C, D, or 1, 2, 3, 4, or he can tell, he can give first three and give the fourth one and the fifth one as X and Y. Then he can ask an added question on the X and Y part, and that's how that question became more complex. Okay, so some rare videos explain very easily. For example, my own video, I'm hopeful that this is a video that can help you very easily. So the mnemonic for the memory to by heart the sequence of the male axillary ducts is some rare videos explain very easily. Some for seminiferous tubules, rare for retate testis, and videos for vasa efferentia, and explain epididymis, and very vast difference and easily is ejaculatory duct. And you know that after ejaculatory duct, obviously it goes through urethra and urethral meatus. So let's move. Key points. Sperms are produced in seminiferous tubules. They mature and get stored in the epididymis. So what is the function of epididymis? Production of the sperm happens in the seminiferous tubules, but they are just stored there to get matured. Storing plus maturation in epididymis. Storing plus maturation happens in epididymis. During ejaculation, sperms move through vas deferens and ejaculatory duct to reach urethra and exit through the urethral meatus. High yield is learn the exact sequence. That is important. Okay, one mistake can lose a mark. So sequence is important, and the memory trick for this male axillary duct sequence is some rare videos explained very easily. Okay. Previous year questions, direct questions on sequence are frequently asked. Now it can integrate. For example, it can ask you what is the fourth in the sequence. Suppose the fourth one is being told as X, then find what is X and what is the function of X. So that is how the question could become a bit lengthier. Sequence plus functional identification question can be asked. Stop the video here, observe the whole slide, and by heart the sequence thoroughly. Male axillary ducts. Additional details, high important directly. Each duct's function can also be asked. Okay, let's see what are those. Epididymis, we have studied earlier. The storage and the maturation of the sperms happen in epididymis. Urethra, shared terminal duct of reproductive and urinary system. So urethra has both parts. Okay, it has shared function of reproduction and urinary. Ejaculatory ductum formed by the union of vas deferens and duct from seminal vesicles opens into urethra. So vas deferens plus the duct of the seminal vesicle gives rise to ejaculatory ductum, and vas deferens receives a duct from a seminal vesicle and opens into urethra as ejaculatory duct. Labeled questions can also can be asked. Okay, memory line you can see here: store, carry, join, exit. Epididymis, vas deferens, ejaculatory duct, urethra. Okay, see what I'm trying to see this do there. Okay, the sequence along with function. First is storage, then is carrying, then it is joining, and then it is exiting. So in that sequence, epididymis, storage, vas deferens, carry, ejaculatory duct, join, and the exit is urethra. So all these questions in the memory based also and diagram based also. And there was a previous question. The shared terminal duct of the reproductive and urinary system is human male. And the expected question now is the duct of a seminal vesicle plus ejaculatory duct is directly opening into urethra. That type of questions can be asked directly. Male axillary glands and seminal plasma. This is out of all these slide, this is high yield, very much high yield actually. Okay, so let's see what are the axillary glands. Okay, glands are paired seminal vesicles. Paired means there are two, one on each side. A prostate and a paired bulbourethral glands, also called as Cowper's gland. So there are three types, total three pairs, and total in number are six. So two seminal vesicles, a prostate, and two bulbourethral, five totally. Prostate is only one. Okay. So prostate is one, and bulbourethral glands are two, and seminal vesicles are two, each one on each side. And bulbourethral glands are also called as Cowper's glands. What is seminal plasma? Fluid part of a semen which is rich in fructose, calcium, and certain enzymes. So the seminal plasma is nothing but the fluid part of the semen. Apart from that sperms, the fluid part is called as seminal plasma, and it is rich in fructose, calcium, and certain enzymes. So moving on, what is the function of seminal plasma? As you can see that the plasma is rich in fructose, calcium, and certain enzymes. So the function is it provides nutrition, it activates sperms, and it lubricates the bulbourethral glands. And collective secretions, all these collective secretions is called as semen. So what are the previous year questions? Let's see. Seminal plasma in human males is rich in fructose and calcium and certain enzymes. Secretions from which one of the following are rich in fructose, calcium, and some enzymes? Male axillary glands. And the high yield topic is axillary glands add volume and quality to semen by providing nutrients, enzymes, and lubrication. Stop the video here, read whole the slide, revise, and see the diagram also so that you can remember directly image-based recollection during the exam. Let's go into the female reproductive system. Okay, obviously female reproductive system parts are very important. Ovaries are the main part, then comes the female axillary ducts. Let's go with ovaries. Okay, you can see the diagram also where they are located. Okay, ovaries, primary female sex organs are called as ovaries, produce ovum, that is a female gamete, and the male gamete is sperm, and steroid hormones like estrogen and progesterone. So the function of the ovum is not only limited to production of female gamete. The function of the ovaries is not only limited to production of a female gamete ovum, but also the function is to produce hormones, that is estrogen and progesterone. The location of these ovaries are on each side of the lower abdomen. Okay, you have to see the diagram also. The diagram in the anatomical body here, so that you can see the location, and also the diagram of the actual ovary. Okay, we are going to see that again. The size of these ovary is 2 to 4 centimeter in length, covered by thin epithelium. Inside is ovarian stroma, divided into cortex, peripheral, and medulla inner. You will see the word cortex, peripheral, and medulla inner in the kidney also. We are going to study that again. Okay. So you can see there is germinal epithelium, there is ovarian cortex, and there is ovarian medulla. Female axillary ducts. Oviducts or fallopian tubes are about 10 to 12 centimeters long. Parts from ovary to uterus. Infundibulum. The first part of these fallopian tubes, which is closest to the ovary, is the infundibulum. What is infundibulum? Direct fact-based questions are asked. Okay. Infundibulum is a funnel shaped closest to ovary. Its edges have finger-like fimbriae. You also see the word finger-like projections in the intestinal villi. Here, finger-like fimbriae that help collect the ovum after ovulation. So, after the ovulation, from the ovary to the fallopian tube, the ovum is collected through this fimbriae, this finger-like structure, that is infundibulum. The second part is ampulla, wider part and site of fertilization. The important site of the fertilization is ampulla. And then comes the isthmus. There is the narrow lumen joins the uterus. So joining part is isthmus. We'll study the part isthmus in the thyroid gland also. That is also having narrow joining point. So infundibulum, fimbriae pick up the ovum released from ovary. You can see the diagram there. The diagram is self-explanatory in nature. Then ampulla, where fertilization occurs in the ampulla, and the isthmus is the uterus with narrow lumen. It joins the uterus with the narrow lumen. You can see there actually in the diagram. What are the high yield facts? Let's see. Ovulation means release of ovum from ovary, okay? And fertilization occurs in the ampulla of the duct. The memory you have to remember as a trick point is I am I. Infundibulum, ampulla, isthmus, uterus, order to remember. Okay, the previous year question which has been asked from this female reproductive system is site of fertilization in humans is ampulla, and the part which helps in correction of the ovum is fimbriae of the infundibulum, and finger-like productions is called as fimbriae. Okay, know the parts in order and the functions, and that is how the questions are asked. The female reproductive system, ovaries are the primary female sex organs. They produce ovum, female gamete, and steroid hormones, estrogen and progesterone, located one on each side of the lower abdomen. Size is about 2 to 4 centimeter in length. Covered by thin epithelium inside is ovarian stroma divided into cortex, peripheral, and medulla inner. Oviducts, there is fallopian tubes, infundibulum, ampulla, and isthmus. These questions have we have seen in the previous slide also. Uterus. The common word for the uterus is womb. Okay, inverted pear shaped. So the shape of the uterus is inverted pear shaped. Layers from outside to inside are one, perimetrium, that is outside, myometrium middle, and the endometrium the inner side. Okay, so there are three layers. PME, from outside to inside. Perimetrium is external thin membranous layer. So peri is membranous, myo is muscular, and endo is glandular. We are going to see that. So perimetrium is an external thin membranous layer. Myometrium is a middle thick layer of smooth muscle, exhibits strong contractions during delivery. So this is made up of muscle. So this muscle have it shows contractions by which the baby is going to come outside. So during delivery, the main function of the myometrium is to exhibit strong contractions, and the endometrium is inner glandular layer. It means it is a made up of glandular structure gland which secretes some things, undergoes cyclical changes during the menstrual cycle. We know that all the female after puberty goes through a cycle of chemical and hormonal changes in the uterus, that is called as menstrual cycle, that happens due to endometrium. Moving on, the next part after the uterus is cervix. So cervix is a narrow part that opens into vagina. So in the lowest part is vagina and the upper part is uterus, and in between those two we have cervix. I mean females have cervix. Narrow part that opens into vagina. Cavity of the cervix is called as cervical canal, which along with vagina forms the birth canal. So birth canal means cervix plus vagina, and the womb means uterus. Cervix, that is a narrow part. Okay, previous year questions, obviously statement based questions were asked in this from this part. Okay, female external genitalia, also called as vulva. So the parts after the vagina, the external part is called as female external genitalia, also called as vulva. Okay, the main parts you can see, you have to remember this diagram also. This is very important even in your mbbs. So I know that sometimes it looks awkward. Obviously, it's a private part to study, but as per the knowledge and as a science students, you should not shy and you should study everything with pure intentions. For mons pubis, cushion of fatty tissue covered by skin and the pubic hair is called as mons pubis. So you should remember that mons pubis is a fatty layer. Okay, and next is labia majora, fleshy fold surrounding vaginal opening. It is made up of flesh. Labia minora, paired folds under the labia majora, and the important part is hymen actually, and the function of the hymen is a membrane partially covering the vaginal opening. Another important part of the female external genitalia is clitoris, tiny finger-like structure at the upper junction of the labia minora. You should remember that during the coitus the hymen is broken down, okay, and it is turned, actually it is torn during the first coitus, that is during the sex actually, but can also be broken by sports or tampons, okay. Hymen is a membrane partially covering the vaginal opening. There is a bigger myth actually going amongst the males, is that absence of hymen is actually a proof of losing virginity. It is not true actually. Presence or absence of the hymen is not a reliable indicator of the virginity. Okay, previous questions you can see here, statement based questions has been asked. You should remember each and every structure, its function, its position. Okay, and previously there was a match the following question also. Stop the video and study the PYQs. Moving on, mammary glands, breasts, okay, paired structures containing the glandular tissue and fat. So, mammary glands, that is breast, consists of two parts. One is glandular tissue, which is helping in secretion of the milk, and the other is fat. Each breast divided into 15 to 20 mammary lobes containing alveoli, clusters of milk secreting cells. Let's stop here. There was one more structure which has lobe, okay, and that was we have studied in the testis. Now here also we are having 15 to 20 mammary lobes. We had almost more than 250 testicular lobes. Here the mammary lobes are 15 to 20. Another important word which is similar in only spelling and sound, but the function and the structure is completely different. That is alveoli. We have studied, or we will study, the alveoli in the respiratory system. The tiny sac-like structures present at the terminal bronchioles of the lungs is called as alveoli. But here alveoli are the cells which help in milk secreting. So this was about the comparison between the testicular lobes and also the mammary lobes, and also the word alveoli here. Moving on, milk stored in the lumen of the alveoli goes into the alveoli, open into the mammary tubules, and then followed by tubules joined to form a mammary duct. The sequence has always been a part of a question. Okay, so let's see that. Several mammary ducts join to form the mammary ampulla. That ampulla connected to the lactiferous duct and gives outside, opens at nipple. So the sequence is important. Pathway of the milk ejection: mammary alveolus, then followed by mammary tubule, then mammary duct, mammary ampulla, then comes the lactiferous duct. The diagram-based question, the labeling can also be asked. What is alveoli could be confusing because the alveoli word has been also used in the respiratory system. Let's see the previous question. The correct sequence has always been asked, so keep on revising. Sometimes he can directly ask the sequence, okay? Or else sometimes he can give the sequence and miss any 1 or 2 points and ask that one point along with added question into it, okay? And another question is about the match the following in the previous year. Stop the video, revise it very well. Gametogenesis. The production of gametes is called as gametogenesis. Male gamete production, spermatogenesis. Okay. Let's see what is the formation of sperms here. The site of the formation of sperms, as we have studied earlier, is seminiferous tubules of the testis. And it begins at puberty due to increased uh, gonadotropin releasing hormone from the hypothalamus. So the production of the semen starts from the puberty. It is because of the production of increased production of gonadotropin releasing hormone GnRH from the hypothalamus. That is a very important point. It is a volatile statement, one can easily forget it. It stimulates the anterior pituitary to release the LH and the FSH. So after puberty, gonadotropin releasing hormone produced from the hypothalamus goes to pituitary, and it makes the pituitary to release the LH and FSH, and that's how LH and FSH have a role in the sperm production, as we have studied earlier also. Hormone regulation we have studied earlier. LH, that is luteinizing hormone, acts on Leydig cells. It stimulates the androgen secretion, that is androgens are hormones like testosterone, stimulate the spermatogenesis. FSH acts on Sertoli cells and it stimulates the secretion of factors that help spermiogenesis, that is maturation. Steps of the spermatogenesis. Let's see. Spermatogonia are two and diploid. It has two sets of chromosomes. It multiply by mitosis. Some spermatogonia grow into primary spermatocytes. Even the primary spermatocytes are also two and that is diploid, have two sets of chromosomes, undergo meiosis 1. That meiosis 1 also called as reduction division. Then it produces two secondary spermatocytes, that is, it is having only one and that is haploid, one set of chromosomes. So spermatogonia diploid, primary spermatocytes diploid, and secondary spermatocyte is haploid. And from spermatogonia to primary spermatocyte, it is mitosis, and from spermatocyte to the secondary spermatocyte, it is meiosis. Each secondary spermatocyte undergoes meiosis 2. It is two spermatids. Thus, one primary spermatocyte yields four spermatids. You can see the diagram here. Okay, so this is where in the seminiferous tubules the spermatogenesis is occurring. Okay, one is multiplication. The spermatogonia is just multiplying 2n by mitosis. Then there is a growth, that is primary spermatocyte. It divides into maturation meiosis 1 into 2, and these two again goes into meiosis 2, that is spermatids, total 4. So there are two maturation steps. So if you see the diagram carefully, the part where mitosis is happening, it is just multiplication. And the part where the primary spermatocyte is growing, that's part of the growth. And the part where the meiosis 1 and meiosis 2 is happening, that is maturation. And then comes the spermiogenesis and spermiation. Okay, let's go again. So one primary spermatocyte gives rise to four spermatids. Spermiogenesis is transformation of spermatids into spermatozoa. No further division, only transformation. So spermiogenesis is transformation. Then comes the spermiation, that is release of the matured sperm cells from Sertoli cells into lumen of the seminiferous tubules. So that is called as spermiation. Stop the video, read the whole statement and just follow the production of spermiogenesis, the sequence and the divisions also. Spermatogenesis versus oogenesis and spermiogenesis, previous year questions. Let's see. Previous year comprehensive challenge. One can observe here that spermatogenesis has some unique features when compared to oogenesis. Let's see. You know that spermatogenesis happens after the puberty. It means it has to be initiated after the puberty only. But when it comes to oogenesis, it is not that, okay? Even before puberty also you can see that, okay? Stop the video here, just go through whole the picture. You can understand the process of production of this spermatogenesis has a lot of checks. One is sequence and another is the checks where the number is going to get double. That number based questions can also be asked, okay? Structure of a human sperm. Okay, there are mainly four parts. Head, it contains haploid nucleus, genetic material. Anterior portion is covered by acrosome, cap-like, filled with hydrolytic enzymes, hyaluronidase and acrosin that help in fertilization. That is how it is going to enter into the ovum. Neck contains centrioles. Middle piece, numerous mitochondria coiled around axial filament provide energy ATP for tail movement. Tail is a flagellum for motility. Okay, so direct fact-based questions can be asked from this slide. Moving on, human sperm. Previous year, you know, what is the middle piece and what does it contain? And what is the function of mitochondria? Okay. And in the same way, what is the function of acrosome? What do you mean by sperm lysine? This question has been asked previously. Match the following has been also been asked. Just stop the video, go through all the video image-based explanations of the PYQ so that even in a new question is been asked, your brain can imagine this image and recollect the data. Oogenesis. After spermatogenesis, now comes the oogenesis. So what is oogenesis? Basically, it is formation of ovum is called as oogenesis. Okay. Let's see what is the key differences between spermatogenesis and oogenesis. Okay. Spermatogenesis starts after the puberty, at puberty, whereas oogenesis during the embryonic development itself, at the fetal state itself, when the baby is inside the mother's womb, At that time itself, if that is a female fetus, then the ovum development has started. Okay, and it has four functional sperms, number of gametes, and here it is one functional ovum plus two to three polar bodies. Meiosis completion continuous completes in testes, whereas meiosis 1 is complete just before the ovulation, and meiosis 2 completes only after the fertilization. Occurrence after the birth continues throughout life, and whereas in the oogenesis it ceases at the menopause. Stop the video here. Try to compare the spermatogenesis and oogenesis. Direct comparison based questions statement form can be asked. Previous year questions, you can see here, at which stage of life the oogenesis process is initiated? Embryonic development stage. In human females, meiosis 2 is not completed until fertilization. True or false statement questions are also asked. Meiotic division of the secondary oocyte is completed at the time of fusion, that is during the fertilization. Exclusion of the second polar body from egg nucleus occurs after entry of the sperm but before fertilization. Capacitation occurs in female reproductive part, not in the oogenesis. So these previous year questions has been asked. Stop the video, revise all the questions. Maybe same question will not be repeated as it is, but it can be integrated and question can be asked. So these are the stages. Stage 1, 2, 3, 4, 5, 6, 7. Let's see step by step. First stage is the formation of primary follicle. Okay. This primary follicle has a primary oocyte. It is nearly 60 to 80,000 are present per ovary at the birth. It has single layer of granulosa cells. Okay. Statement based question was asked. Okay. Secondary follicle, more layers of the granulosa. If only one single layer of granulosa cell is present, it is primary follicle. And the second stage is more layers of the granulosa. So cloning multi-channel pipette are used in order to identify the secondary follicle. Tertiary follicle, obviously you can see that the primary oocyte grows and completes the meiosis one. There is secondary oocyte and there is also a fluid filled space that is called as antrum. If these three parts are can be seen in one stage, that stage is called as tertiary follicle. It has also some parts called as theca interna and theca externa. Then comes the Graafian follicle. It is a mature tertiary follicle. Okay, it is called as Graafian follicle. You can see here it completes the meiosis 1, secondary oocyte, and the meiosis 2, but it is arrested at the metaphase 2. The meiosis 2 is started but not completed. Okay, and it is being arrested at the metaphase 2. Direct memory based questions have been asked from these one slide. Then comes the ovulation. Rupture to release the secondary oocyte is called as ovulation. Okay, you can see from the Graafian follicle, this secondary oocyte is being ruptured. Okay, then comes the corpus luteum. Okay, so what is corpus luteum? Basically, the remnant Graafian follicle is called as corpus luteum, essential for maintenance of the endometrium. Okay, then is corpus albicans. It is corpus luteum identification. After that one, what remains is corpus albicans. So previous, stop the video here. You can study all these important PYQs. Yeah, at each state, at each stage, I have given you one previous year question so that stages are important and the questions are asked not only as a sequence part, but what happens at each stage is also very important when it comes to ovulation. Menstrual cycle. It is actually very easy. You just have to remember each phase and the function and also the hormonal changes happening. Definition. Reproductive cycle in the female primates, monkeys, apes and humans is called as menstruation cycle. First menstruation happens at the puberty is called as menarche. Cessation, that is stoppage, is happening at menopause. It is approximately 50 years. For some moments it is 40, 41, 42, 45 up to 50. Average cycle length is 28 to 29 days. One menstruation cycle extends till 28 or 29 days, approximately 1 month. Let's see the phases of the menstrual cycle. Okay, what is the phase and how many days does it happen? What are the events happening in that stage of the phase and what are the uterine events, and what are the hormonal changes. So direct statement-based, fact-based questions can also be asked, and also the match the following from the table. Let's see. Menstrual phase in the starting phase, it lasts for 3 to 5 days. Minimum 3 days, maximum 5 days. And the ovarian events are low estrogen and progesterone. What happens in uterus is breakdown of the endometrial lining, menstrual flow starts, and withdrawal of the progesterone happens when it comes to the hormonal changes during the menstrual phase. Follicular phase, also called as proliferative phase, it is 5 to 13 days it lasts into that time period. And what happens in this one is in the ovaries, primary follicles converts into Graafian follicle, and increasing of the estrogen is also seen in the follicular phase. So primary follicle to Graafian follicle, follicular phase. You can easily understand that one, okay? And what happens in the uterus is endometrium regenerates. So therefore, proliferation of the endometrium happens. That is why this follicular phase is also called as proliferative phase. What are the hormonal changes happening in the second stage, that is the follicular phase, is FSH and LH increase gradually, estrogen rises. Next comes third stage is ovulation. Ovulation happens exactly and approximately by the 14th day. Okay, rupture of the Graafian follicle and release of the secondary oocyte happens in this ovulation phase, and the hormonal changes is LH surge, that is luteinizing hormone surge is peak highest at this one. It induces ovulation, and FSH also peaks. Highest volume is present at the on the day 14th of menstrual cycle. Luteal phase, also called as secretory phase. What happens here is nearly it lasts for 15 to 18 days, 28 days. So this luteal phase, what happens in this luteal phase is ovarian events. Remaining follicle transforms into corpus luteum. In the uterus, endometrium becomes secretory thickened because of the progesterone effect, and high progesterone from the corpus luteum is produced as part of the hormonal changes. Previous questions, a lot of questions I'm displaying here on the video. Stop the video and read the previous question and link it with the previous slide I have shared. Fertilization and zygote formation. We know that fertilization happens in the fallopian tube. Okay, what is required for fertilization? Obviously, we require ovum from the female and sperm from the male, and though both are transported simultaneously, no. One primary, it is not actually, okay. It comes differently. Simultaneously doesn't happen. That was a part of the PYQ. One primary oocyte completes the meiosis one. The events of fertilization are related with the structure of the sperm. Okay, sperm reaches the zona pellucida, and the sperm is entering into the ovum. It is going to pierce the zona pellucida. How is going to pierce? Because of the acrosome reaction. Acrosine and hyaluronidase are the enzymes present in the acrosome. It is going to help penetrate the zona pellucida, and then comes the cortical reaction. Zona pellucida becomes hard, and that is how many sperms will not enter. Only one sperm, as soon as one sperm enters, and that zona pellucida becomes hard, so that in one ovum only one sperm can enter. After that, when there is completion of meiosis 2, sperm entry induces the completion of meiosis 2 of the oocyte. Secondary oocyte arrest at the metaphase 2, but after the entry, meiosis 2 is complete. Then, second polar body, haploid ovum, also called as ooted, you can see there. Then the male and the female pronuclei fuses, and so after successful and maturation, then that is how it forms the diploid zygote. After that, when human embryonic development is an important thing, okay, cleavage to implantation. Previous year questions, integrated image has been presented here. Let's see what happens. What is cleavage? Cleavage is actually cell division and formation of multiple cells, okay. So what happens is after the formation of a zygote, it goes into rapid mitosis. That rapid mitosis is called as cleavage. So zygote goes into 2 cell into 8 cell, and that cell number increase. The increased cell number is called as blastomeres, and at the level of 16 cell, it goes outside. It goes through the duct, and that is convert into morula. That is 8 to 16 cell stage is called as morula, and then zygote, cytoplasm, DNA, all things are internally developing. Okay, so there was a previous question. Which statement about morula is correct? It is has almost equal quantity of cytoplasm as an uncleaved zygote, but much more DNA. Yes, 16 times more DNA. That is happens in morula. Second stage is blastocyst and anatomy. What is blastocyst? Okay, so the blastocyst is actually a differentiated and mature form of those cells. What do you mean of differentiation? Differentiation means all those cells which are divided and they are present in the morula, those cells are differentiated into layers. From one layer, one germinal uh, layer comes, like mesoderm, endoderm, and ectoderm. And from each layer, different organs are produced. Such type of a stage where you can see cells being differentiated and mature is called as blastocyst. Okay, so trophoblast is outer layer will form placenta. Inner cells, that is embryoblast, will form the embryo. Then comes the implantation. What is implantation? Basically, the implantation is fixating of that stage of the blastocyst into the endometrium. It happens after the 6 to 7 days after implant fertilization. So in human female, the blastocyst gets implanted in endometrium by the trophoblast cells, because trophoblast cells are outer layer. Okay, so stop the video here, study this image specifically, take out time. Direct questions will be coming from this slide. Next comes sex determination. It is also a part of genetics. Here we are going to study an overview. Okay, so female gamete always carries X chromosome. Male gamete carries X or Y chromosome. If X-bearing sperm fertilizes the ovum, then the female is produced, and if Y-bearing sperm fertilizes the ovum, then the male is born. Therefore, sex of the baby is determined by the father, not the mother. Let's see the diagram here. You can see the diagram. Stop the video. You can see that ovum is always XX and the sperm is X or Y, and that's how XX female and XY male. Previous year question. Okay. In our society, women are blamed for producing female children. Choose the correct answer for this determination. Due to the genetic makeup of the particular sperm which fertilizes the egg, therefore, actually male is the responsible for the sex determination, not the female. Stop the video and study the PYQs. Okay, key points. Sex is determined by the sperm and not by the ovum. Sperm binds to ZP3 receptors on the zona pellucida. Okay, and once it penetrates inside the zona pellucida, it is becoming more stronger and it is impenetrable. Acrosome reaction triggered by lysine enables the sperm to penetrate the zona pellucida. Then fertilization occurs in the ampullary isthmic junction of the fallopian tube, or just ampulla is also the correct answer. Pregnancy and embryonic development. What is placenta and what are the functions? Let's see. Okay, formed from the chorionic villi, that is trophoblast finger-like projections interdigitated with uterine tissue. So what is placenta? Basically, placenta is coming from the trophoblastic layer, the finger-like projections of the trophoblast after the conversion into the blastocyst, after implantation. We know that blastocyst is going to get implanted in the uterus through its what you call a trophoblast layer. That trophoblastic layer, finger-like projections, that is going to go inside the uterine tissue. That is how the placenta is formed. That such type of finger-like structure is called as chorionic villi, connected to the embryo by the umbilical cord. Functions. What is the functions of placenta? So functions are supply of oxygen and nutrients to embryo, removal of carbon dioxide and waste from embryo, endocrine organ, it secretes human chorionic gonadotropin, maintains the corpus luteum during early pregnancy, human placental lactation, growth of the mother and breast, estrogen and progesterone. All these are produced by the placenta itself. Okay. Later phase relaxin is also secreted by the ovary, that is corpus luteum. Remember the structure of the placenta, the upper part, uterine tissue, the penetrated part coming from the chorionic villi. What is chorionic villi? Chorionic villi is the trophoblast projection. What is trophoblast? Trophoblast is the outer layer of the blastocyst. This intervillous space in the maternal blood, and it is connected to the embryo via umbilical cord. Let's see the functions. The functions are divided into three. One is transport, other is endocrine, that is secretion, and the third is protective. The transport function of the placenta is supply of oxygen and the nutrients to the embryo and removal of the carbon dioxide and the waste from the embryo. The endocrine, that is secreting part, is human chorionic gonadotropin and human placental lactogen and estrogens and progesterones and later phase relaxin. The functions of HCG, HPL, estrogen, progesterone, and relaxin is important. Direct questions can be asked, and the site of production can be asked, and also the match the following. Key points. Placenta is formed from chorionic villi interdigitated with the uterine tissue. It connects the embryo to mother by umbilical cord. Placenta is a transport, endocrine, and productive organ. Three functions of the placenta are very important. It secretes HCG, HPL, estrogen, progestogens, relaxin, and placenta does not secrete oxytocin. Remember, that could be a confusing question. Stop the video, study all the pre-YQs so that you can integrate the PYQ with the content present in the slide. Human embryonic development timeline. What happens in the first 3 months, what happens in the second 3 months, and the what happened in the last 3 months. As we know that the gestation period of the embryonic development in the human being is 9 months approximately. Actually 9 months, 7 days. But we can divide it into 3 months quarterly. That is called as first trimester, first quarter, second trimester, third or between second or the middle quarter, and the last trimester, that is the last quarter. What happens in the first trimester? Okay, in the first trimester, after 1 month, fetal heart is formed. Heart is formed after 1 month of gestation. It is a PYQ. In the end of the second month, limbs and digits develop. Limbs and digits, it means fingers are developed. It is also a PYQ. All the milestones, all the process of the embryonic development, I have integrated each step with the previous year question. Okay, and at the end of the first trimester, that is third month, most major organ systems formed. External genitalia is also developed. It means sex has been finalized. Major organ systems at the end of 12 weeks. In the mid pregnancy milestones, it means the second trimester. In the fifth month, first movements and appearance of hair usually observed in fifth month. The baby starts moving in at the time of the fifth month, and fetal movements are seen. Appearance of hairs is seen in the fifth month. Okay, at the end of the 24 weeks, that is second trimester, development check, body covered with fine hair, that is lanugo hairs are seen, eyelid separate is seen, and eyelashes are formed. Yes, obviously incomplete, it is not complete development. Okay, then comes the final development and quantitative success, quantity success. Okay, ninth month, a fetus is fully developed, ready for delivery, and PYQ questions are has been asked here. Correct statement like major organ systems 12 weeks, heart 1 month, limbs and digits 2 months, hair 5 months. Okay. Parturition and lactation. What is parturition? Childbirth is called as parturition. Gestational period is 9 months, about 40 weeks. Process is vigorous contractions expel the fetus. We have studied in the structure of the uterus that the upper layer of the uterus is perimetrium, the middle layer is myometrium, and the inner layer is endometrium. The middle layer, myometrium, is made up of muscles, and that muscles are going to produce a muscular contractions during the childbirth. Fetal ejection reflex signals from fully developed fetus and placenta induce mild uterine contractions. This triggers the release of oxytocin from the maternal pituitary posterior lobe. Oxytocin causes stronger uterine contractions that gives rise to positive feedback, that gives rise to more oxytocin, that gives rise to the expulsion of the baby and then placenta. See the diagram there. Okay, there is a uterus, and in that uterus there is a baby, a fetal baby lying, and behind that fetal baby there is a placenta, and that placenta is connected to the baby through umbilical cord. Okay, what happens in the sequence is, let's see, fully developed fetus, yani the baby itself and the placenta, both are going to give the signal. It means the blood supply structure is placenta and the baby, both are going to give the signal, and mild uterine contractions are coming from the after the secretion of the placental oxytocin. Then oxytocin released from the placental pituitary gives rise to stronger uterine contractions, then more oxytocin released, then the expansion of the baby, and then placenta is also expelled. Then comes the stage of lactation. What is lactation? Milk production and ejection is called as lactation. Okay, galactopoiesis. Prolactin from the anterior pituitary stimulates milk production by mammary glands. Milk ejection is let down reflex. Suckling by the baby, sensory impulses, hypothalamus, that impulses from the sucking of the baby goes to hypothalamus. Then hypothalamus releases oxytocin from the posterior pituitary. Then the contraction of the myoepithelial cells produces the milk ejected reflex into the duct and out through the nipple. Prolactin is for milk production. Oxytocin is for milk ejection. And there is also a sequence of process: baby sucking, then sensory impulses to hypothalamus gives rise to oxytocin released from the posterior pituitary, and contraction of the myoepithelial cell that gives rise to the milk ejection. Key points to remember is parturition is initiated by the fetal ejection reflex and mediated by the oxytocin. Oxytocin causes strong uterine contractions, parturition and milk ejection lactation. Both the role. Oxytocin has both role. Both the parturition role is also, and also the role of lactation. Prolactin is for milk production and not for the initiation of parturition. Stop the video and study the PYQs and integrate these PYQs with the side content in the slide. Now let's go into what is lactation. Okay, the first milk produced during initial few days after the birth is called as colostrum. Colostrum contains IgA antibodies. Even though you study this one for like tens of times, you are going to forget it because we are going, that is such a dry topic actually. So this image should be imprinted in your brain and it will help you recollect the data during examination. So antibodies which are higher in content in the first milk, that is colostrum, is IgA antibodies. Breastfeeding is recommended for a healthy baby. PYQ. Okay, you can see there colostrum is rich in fat, false, it is rich in antibodies. Oxytocin is secreted by the anterior pituitary, false, it is secreted by the posterior pituitary. And relaxin is secreted by the placenta, false, it is from the ovary or the corpus luteum in the later stage of the pregnancy. Frequently confused terms actually. Spermatogenesis, entire process of the sperm formation from spermatogonia to spermatozoa is called as spermatogenesis. Stop the video, just see all the sections we have already studied, and that's how you can revise and you need not to get confused. Let's see what are the questions predicted. According to the previous year questions, according to the weightage also, the expected questions are: one, assertion reason. The scrotum is located outside the abdominal cavity. We have studied in the earlier slide also, because the outside the body cavity, the temperature is lower, and that is important for spermatogenesis. Okay, sequence based arrangement, I have told you the diagram based questions can come. Match the following of the cells and functions. Statement based questions about the oocytes and LH and other hormones. Diagram based questions of the sperm. and numerical based questions from the one primary spermatocyte gives rise to four spermatocytes, and one primary oocyte gives rise to one secondary oocyte. Okay, so these questions are expected in the future NEET. Stop the video and study it thoroughly. NEET UG, now comes the reproductive health and complete exam notes. This is the only video you need when it comes to reproductive health, and all the questions are expected from this video only. Most highly probable high weightage topics are population explosion and birth control methods like natural method, barrier, IUDs, pills, implants, and surgicals. We are going to study that. Okay. What type of questions match the following are expected? Correct, incorrect statements are expected, and identification of the IUD types with diagrams and their functions are also expected. Second important is infertility and assisted reproductive technology. The third important is reproductive health and sexually transmitted diseases and lower level, but sometimes questions are asked from the medical termination of pregnancy. All these things we are going to cover in this video. Reproductive health problems and strategies. Okay, what is the definition of reproductive health? Okay, reproductive health means a total well-being in all aspects of reproduction, physical, emotional, behavioral, and social, for both the partners, that is male and the female. What are the India's initiatives? Family planning program initiated in the 1951, first country in the world to launch such a family planning program. Family planning means how many children should a couple have. Okay, reproductive and child health care RCH programs currently in operation covering wider reproduction related areas. Important strategies or actions: creating awareness, audio, visual, and print media, sex education in the schools, information about reproductive organs, adolescents, safe sexual practices, STDs, that is sexually transmitted diseases, birth control options, care of pregnant mothers, breastfeeding, equal opportunities for male female child, and providing the medical facilities like pregnancy, delivery, STDs, abortions, contraception, menstrual problems, and infertility. All comes under the reproductive health. Also include a statutory ban on the amniocentesis for sex determination to check the female feticides. It is common in India that all the families and all the couples expect their child to be male, and there was a huge level of crime happening, that when we know that like many people identified that their child is actually a girl, and they started killing inside the womb, or at least after the birth of the that female fetus, that is called as female feticide. So there was a ban on sex determination, and the process by which this sex determination happens is amniocentesis. So let's see what is amniocentesis. Amniocentesis, there is an amniotic layer and there is an amniotic fluid. Okay. Amniocentesis is allowed only to detect genetic disorders, Down syndrome, hemophilia, sickle cell anemia, and fetal survivability. Is not a technique for sex determination in legal practice. The fourth important part of the reproductive health is immunization of the children, and the fifth is research and development. Saheli is a new oral contraceptive for females developed by Central Drug Research Institute, Lucknow, not ICMR. It is developed by CDRI. Saheli is new, it is an oral contraceptive tablet actually. The name is Saheli. Previous year question, stop the video, connect the topic and the question directly so that you can tell what is happening in India. So, what is examination prep? Amniocentesis is banned in India is a false statement. The sex determination by amniocentesis is banned, but amniocentesis is not a totally banned one. Okay, Saheli is a non-steroidal pill. It is not a steroidal pill. Population explosion and birth control. Let's see the population statistics as per the NCRT. World population as per the 1900 census is 2 billion. Expected by 2000 is 6 billion, and by 2011 is 7.2 billion. Now it could be 7.6 billion. India, it has nearly 350 million at independence and 1 billion by 2000, and it has crossed 1.2 billion by May 2011, and it has crossed 1.4 by now. Population growth rate is less than 2%, but still it is rapid. Reasons for the population explosion is decline in death rate. Healthy conditions declines the death rate. Maternal mortality rate also declined. Infant mortality rate increase in number of the people in reproductive age. Okay, so all these are the factors of the reasons for explosion. Stop the video and by heart those sentences because direct statement based questions are expected from this topic. How do you check? How do you control the population growth? Motivate smaller families using contraceptive methods. Statutory raising of the marriageable age for female 18 and for male 21. Incentives for small families, that is gifts and slogans like Hum Do Hamare Do could help public to stand understand that we how to maintain the population. The ideal contraceptive, what is ideal contraceptive and what are their properties? Okay, what is an ideal or perfect way to adapt an contraceptive? What is contraceptive? Contraceptive means that structure or that part which is going to stop the fertilization to happen. Intercourse or sex can be done, but fertilization won't happen. It means either it will stop the sperm to go out, or it will stop the sperm to go and meet the ovum. Both the way it will be a contraceptive only. Conception means fertilization, and contraceptive means how it is going to stop the conception. So how to identify the perfect or ideal contraceptive? The properties are it should be user friendly, it should be easy to use, it should be easily available, it should be effective, it should be reversible, and it should have no or least side effects. It should not interfere with the sexual drive or act. Okay, we are going to classify the contraceptive methods also. It can be broadly classified into natural and other barrier effects. We are going to study in the next slide. What is natural methods? Periodic abstinence. It means there is a time period in the menstrual cycle where during that time period, if a couple goes into intercourse, into sex, then that time period there will not, the chances of fertilization are least. Okay, avoid coitus from day 10 to 17 of the menstrual cycle. That time period is called as fertile period. Reason, because ovulation is expected around day 14, and sperm can stay from 5 days to 7 days inside the female body, and that's how it is from 10 to 17 or days of the menstrual cycle. Withdrawal, that is coitus interruptus. It means exactly during the time of ejaculation of the sperm, the male can come outside during the sex. That is such a uh, like withdrawal effect of the uh, intercourse performance. Then comes the lactation amenorrhea, absence of menstruation and ovulation during intense lactation following parturition, effective only up to maximum 6 months after delivery and only if mother breastfeeds fully. Lactational amenorrhea is effective for 2 years. False statement. Only for 6 months. Withdrawal method has zero failure rate. No. Even after the withdrawal, even a drop of the semen also has shown a successful fertilization. Barrier methods are usage of condoms. Male condom, commonly known as Nirod. It is a rubber latex sheet over penis. Also protects the sexually transmitted diseases and AIDS. And for females, there is a female condom that is inserted into vagina. Diaphragms or cervical caps and valves are also used. Rubber barriers inserted into the female reproductive tract to cover cervix. They are reusable. Spermicidal sperms are dying off because creams and jellies or foams are often used along with them to increase the efficiency. Okay, so previous questions have been asked here. Copper releasing intrauterine device, non-medicated intrauterine device, contraceptive barrier, hormone releasing intrauterine device. All these match the following questions are previous questions. We are going to study these words and these details in the further slides. Oral contraceptives. What do you mean by oral contraceptives? Oral, it is taken by mouth. It means it has to be a tablet. Contain either progestogens alone or progestogen estrogen combinations. So these oral contraceptive pills, commonly known as pills actually, or birth control pills, these are of two types. Either just progestogen pills or progestogen plus estrogen pills taken daily for 21 days starting within first 5 days of the menstrual cycle, then 7-day gap menstruation occurs, and then that has to be repeated 21 days pills. Okay, mode of action, what does it do? It is going to inhibit the ovulation and implantation, alter the quality of the cervical mucus to prevent the or retard the sperm entry. And Saheli is a non-steroidal once a week pill developed by CDRI India. It is one of the successful and most commonly used emergency contraceptives is administration of progestogens or progestogen estrogen combination or IUDs within 72 hours of the coitus to avoid the pregnancy due to rape or unprotected intercourse. Combine this topic of the OCPs with the previous question, you will be perfect to answer any question related to this topic. Injectables and implants, okay? Progestogens alone or with estrogen injections are also available, or these implants on the skin are also available. The implant on the skin is going to release inside the body, and that's how it is going to act for a longer period of time. Mode of action is similar to pills, and it has a longer effective period. Previous question, non-medicated IUD is lipase loop and copper releasing IUD is multi-load 375. Hormone releasing intrauterine device is LNG20 and implants are progestogens. You have to by heart this table, this match the following 100% expected questions from here. For males also there are a problem. There are some methods of stopping the fertility. Surgical methods called as sterilization. Terminal method, poor reversibility, vasectomy. Okay, in male, small part of the vas difference is removed or tied, blocks the gamete transport. Sperms produced but not ejaculated. Semen has no sperms. So a male can participate in the sex, but that semen does not have any sperm. It has only fluid. Okay, you can attach that topic with the PYQ. Tubectomy in the female also, the tube can be tied, a small part of the fallopian tube or removed or tied. That is done through abdomen also, through vagina also, and that's how fertilization can be stopped. Okay, medical termination of pregnancy, intentional or voluntary. It means it is happening by the will of the human being. Pregnancy before the full term, before the whole baby is developed, is called as induced abortion, legalized in India in 1971 with strict conditions. Okay, what is the safe period? The safe period is first trimester. यानी पहले के 3 महीनों में ही अगर आप टर्मिनेशन यानी अबॉर्शन करते हैं तो वह सेफ है. Second trimester abortions actually are riskier and associated with more complications. Misuse. This is being misused because once if somebody identifies, the baby inside the womb is a female, and there is a huge type of societal behavior that female babies are rejected and then they will go into abortion. That is the misuse of the medical termination of pregnancy. The Medical Termination of Pregnancy MTP Act 2017 tells that up to 12 weeks, opinions of one registered medical practitioner is mandatory for you to go into abortion. And from 12 to 24 weeks, opinion of two registered medical practitioners is mandatory. And the ground should be risk to the life or grave injury to the physical or mental health of the pregnant woman should be assessed. And substantial risk that the child would be seriously handicapped should also be assessed. Sexually transmitted diseases. Okay, it means the diseases which are transferred from one body to another body, that is from male to female, the female to male, through direct, obviously, sex or intercourse, and some other procedures also. Common STIs are gonorrhea, syphilis, genital herpes, chlamydiasis, genital warts, trichomoniasis, hepatitis B, HIV, AIDS. What is gonorrhea or bacteria? Gonorrhea is caused by a bacteria. Syphilis is caused by a bacteria, and Genital herpes is caused by a virus. Chlamydiasis is caused by a bacteria. Genital warts is caused by HPV virus. Trichomoniasis is caused by a protomonas and protozoan. And hepatitis B is caused by a virus, and HIV/AIDS is caused by a virus. Modes of transmission is most commonly sexual intercourse primarily. Sharing of the infected needles, surgical instruments. For example, any a doctor, mostly an RMP doctor in the villages, in the remote areas, is going to use a single urine for a syringe for many patients, and that's how if one has a disease with the same usage of the syringe, the disease can be transferred to the another patient. And through blood transfusion, if it is not cross-checked and cross-verified, the blood transfusion can also transfer these viruses and bacteria. Infected mother to the to the baby also for hepatitis B, HIV, and genital herpes. Curable versus non-curable STIs. Curable, completely curable if detected early. That is gonorrhea, syphilis, chlamydiasis, and trichomonas and genital warts. Okay, but some strains can be considered curable as per the NCRT. Not completely curable are genital herpes viral and hepatitis B and HIV, all the three. Except hepatitis B, genital herpes and HIV, others are curable according to the NCRT. Stop the video and study the previous year questions and integrate that topic with the slide so that you will be perfect to answer all the questions when it comes to STIs. Complications of STI. What happens if somebody is not treated with the sexually transmitted disease? ऐसा कोई disease अगर आ जाता है और वो for example treatment नहीं होता है, then that will develop into complicated one. Complicated. That is called as complications of STIs. What happens is pelvic inflammatory disease. It is going to spread into the other female reproductive organs like uterus, fallopian tubes, and ovaries. There is increased risk of miscarriages and abortions. Infections may lead to fetal death. Fetus will develop, but it will develop after the delivery. It is died. And ectopic pregnancy. It means pregnancies happen not in the uterus but in the fallopian tube. That is also because of the sexually transmitted diseases. Infertility, damage to the reproductive organs can lead to infertility. That it means that woman cannot have more child or at least one child. Cancer of the reproductive part also a part of the complication of the sexually transmitted disease. Moving on, what is the prevention? Yes, obviously avoiding the sex with unknown and the multiple partners is mainly important. Next is using of the condoms, and early detection and treatment by the doctors is very important. Okay, untreated STIs can cause severe health problems and can also be transmitted to the baby during pregnancy. You should remember that one. Infertility and assisted reproductive technologies. For example, a couple after the marriage is not having any children, then that couple can be like infertile or subfertile, or there is a problem with their fertility that is called as infertility. Inability to conceive or produce children even after 2 years of unprotected sexual cohabitation is called as infertility. So you can only tell that the couple is infertile after seeing 2 years. The causes are physical, congenital, diseases, drugs, immunological, and psychological. Often the male partner may have a problem, not always the female. Then comes the assistant. It means if those couples after 2 years also they don't have children, then medically we are going to assist them to have fertilization and children. These are the techniques that help infertile couples to have children. How do they do that? One, in vitro fertilization. Test tube baby program. It means ova from wife or donor are collected, then sperm from the husband donor are collected. Ova and the sperms are induced to form a zygote in the laboratory, that is in vitro. Zygote divides to form embryo. The embryo is transferred. Embryo transfer, ET is done. Types of embryo transfer, okay, that is according to the site also, stage also, okay. Zygote or early embryo transfer up to 8 blastomeres. Site of transfer is fallopian tube. It is called as zygote intrafallopian transport ZIFT. An embryo with more than 8 blastomeres are transferred to the uterus. That is called as intrauterine transfer. Test tube baby or in vitro fertilization means fertilization of ovum by sperm outside the body in glass and then implantation of the embryo in the female reproductive tract. Only up to embryo only it happens in the lab, and the remaining from the embryo to the fetus happens in the reproductive tract, that is uterus. Stop the video and study the previous year questions and integrate that questions with the content. Then comes the gamete intrafallopian transfer. Transfer of the ovum or the gamete from the donor into the ovum into the fallopian tube and fertilization in the body. But the gift happens, the fertilization happens in the body in the gift. Then comes the ICSI, that is intracytoplasmic sperm injection. It means a specialized procedure where a single sperm is directly injected into the ovum in the lab. Then the single sperm is selected, then it is injected in the ovum, then embryo is formed in the lab only again. Okay, then the ET happens, that is embryo transfer. Assisted reproductive technologies ART are used to help the fertile couples to have children when natural conception is not possible. Stop the video and study the whole slide along with the PYQs. Artificial insemination AI or intrauterine insemination. It means semen collected from the husband or donor is artificially introduced into vagina or the uterus, used when the male partner cannot inseminate or has low sperm count. So the reason for the AI, that is artificial insemination, is problem with the male partner. Okay, what is the process? It is very simple. You can see the diagram. Okay, then the last option is adoption. There is a legal adoption also. Okay, stop the video and integrate that whole topic with the PYQ. These are some of the concepts which are considered to be rare and lesser important, but most of the time questions are asked. So stop the video and read each question, its answer, its mechanism. 100% question is expected, at least one question is expected from this slide. These are all some of the important topics. Stop the video and just study each and everything. These are some of the predicted questions, that is expected questions that can come in our next future Neet. Few questions are assertion reason, few are match the following, few are statement based, few are sequence based, few are diagram based, few are numerical based. We have studied in the previous slides also. Frequently confused terms, sometimes direct term and term definitions can also be given that, and in that, most of the times we get confused and we match it with the wrong term, or many of the terms are closely enough to get confused. So stop the video and just study this one. Obviously, memorize the all the type of IUDs, non-medicated IUD, copper releasing IUD, and hormone releasing IUD, and know which STDs are curable and not curable. Understand the difference between ZIFT, IUT, and GIFT. What is a Saheli? What do you mean by amniocentesis? Natural methods of contraception, all the things you have to understand. I think obviously if you study all my videos, 360 out of 360 in biology is compulsory, hopefully. So stop the video here, revise this video into 2x speed and watch this video multiple times. Don't look at the video, my video on screen, just see the slides and that will be helpful enough. Let's meet in the next video. Thank you. Subscribe to my channel and share to your friends.